So this is part 2 of chapter 5, and it is about the chromosomes and inheritance. And this is the XY chromosomes, the XX for females and XY for males. Okay, so if you look at this, what you're seeing here is called karyotyping. Karyotyping is a photograph of the chromosomes, and they can actually do it for you. They can take a picture, a photograph of your chromosomes, and they can put it together like this under the microscope. This looks kind of very close to an actual picture under the microscope. It looks like this. Okay, so can you tell me how many chromosomes in this?
23.
23 chromosomes or 23 pairs?
23 pairs.
23 pairs. So the total is
46.
46. Is that male or female?
Female.
What do you guys think?
The female.
You can see here, if you can see this one. This is a female. How did you know? Which one determined male or female?
The last one, 23rd pair.
Number what?
23.
23. It's number 23 that determined. So you leave all of this and just look at this. The last one. And the last one, the last pair of chromosomes is the one that can tell you if this is male or female, and obviously this is a female. Okay? Why do we need to produce sperm and eggs, which is gametes? Because the sperms and eggs, which is the gametes, contain haploids, and when they merge together, they make a diploid. If that was not the case, merging will not make a normal number. So we have 23 from male, 23 from female. When they join together to make the zygote, it contains 46, which is okay, which is normal. If that was not the case, and there are no sperms or something, you will have like 46 and 46, and that will be double, and this is abnormal. And sometimes there are abnormalities that can have one of those that's unusual. So this is for the haploid and diploid. Hapl being half, double meaning diploid meaning double. So haploid is 23, diploid is 46. Okay? In order to produce the gametes, are the gametes haploid or diploid?
Haploid.
Haploid. So in order to do that, you need to use meiosis. Meiosis. Do you guys remember when I talked about the translation of mitosis versus meiosis? Does anybody remember what mitosis means? What's mitos? Translation of mitos. Threads. Threads. Mitos means threads. The spindles. Do you remember those spindles that attach the chromosomes and move them away?
Oh, yeah.
These spindles or threads means mitos. So the mitosis is the process of fusing the mitos, and the mitos are these threads or spindles. Is that okay? So now you get it. You double it and then you divide. So you start with 46. You end up having how many with mitosis? Mitosis. You start with a cell that have 46. You end up having two cells. Each one contain 23 or 46. 46. Mitosis is 46. 23. Aren't you decreasing the number from 46 to 23? Reducing the number. Meiosis translation into reduction. Meiosis means reduction. So which one? You produce 23 from 46. Is that reduction? 23 from 46, right? So it's meiosis. Can you remember this? Mitosis, mitos, threads, spindles, mitos. You start with 46, you end up having 46. And this is for the normal cells, the autosomes, the autosomal cells, which is everything except sperms and eggs. Okay? Meiosis is something unique for the sex cells only, for the reproductive cells. And meios means reduction. I start with 46, I end up having 23. Okay? Mitosis had different stages, and we talked about this. Do you guys remember the stages? What's the first stage of mitosis? Here is the interphase, and the interphase is gone. I'm going to mitosis. What's the first one? Prophase. Prophase. Do you remember this when the envelope disappear? Okay, and then the spindles start to form. What's the next one?
Metaphase.
Metaphase. When you arrange along the equator or along the plate. And what's the next?
Anaphase.
Anaphase. And this is when you start to move apart. And the next one is telo, when you moved away. Telo means away, right? And then you form two cells and they move away from each other, and you call that cytokinesis. So you have different stages. It's not exactly the same in meiosis, but it also happened in different steps. And here are the steps. You duplicate during the interphase just like the other one. Here we have just meiosis 1 and meiosis 2. Meiosis 1, meiosis 2. Reduction 1, reduction 2. And remember, you start with one cell. Okay? Having 46, end up having 4 cells. Each one contain how much? How many? 23. 23. You start with 146, end up having 4, 23. And mitosis, you start with 146, end up having 2, 46. Like my skin cell, make another skin cell, make another skin cell. 46, 46, 46. There is no need for reduction for meiosis, but there is a need for reduction when we're talking about sexual reproduction, right? So now remember that the 46 chromosomes are arranged in 23 pairs, right? According to what? Why this come with this as a pair? Because those two are very close to each other, and we call that homologous. So the two pairs, the two, I mean the pair is 2, right? Which 2? Homologous. 2 homologous. This and this very close to each other, and they come together. And that's why even if you look at 23, why this X with X? Because they are homologous, right? Why X with Y? Both of them are 6 chromosomes, right? And that's why they come together. So remember this, homologous. Homologous. So they match together because they are homologous. Okay? So you are going to go, instead of what we did before, you're going through meiosis 1 and meiosis 2. Okay? How does that happen exactly? Here are the stages, or the steps. Homologous chromosomes pairs line up. This is similar to what? In mitosis. When you line like this. Metaphase. Yes, metaphase. And then homologous pairs separate. Similar to what? Anaphase, right? Then you have cytokinesis. So isn't it different? It is different. You have here metaphase, and it's not called that. It's similar to, okay? Anaphase, metaphase and anaphase. And then you have the cytokinesis. And then the chromosomes are going to condense and line up again for the second round. Again. And then condensate and line up along the equator, and then they are going to separate for the second time, and cytokinesis for the second time. So you're doing the meiosis twice. That's what it is. Okay? First you duplicate, but this have nothing to do with this. You duplicate before meiosis. Clear? You duplicate first before meiosis. This have nothing to do with meiosis. Now I'm going to meiosis. 3 steps, meiosis 1, same 3 steps, meiosis 2. Okay? Line up, separate, cytokinesis. And then what? Line up, separate, cytokinesis again. So meiosis 1, meiosis 2. If this is clear, so you have 6 steps, but this is 3, and then you repeat. Unlike the first one, the 6 steps, and when we did in mitosis, these are different things. Any questions so far? Clear? Okay, if that's clear, answer this. Where does it occur? Meiosis versus mitosis. Which cells?
Meiosis, sex cells, gametes, and mitosis is.
So this is 6 cells,
yeah.
and this?
All the other cells.
body cells, any body cells, right?
That's reproduction.
When is it needed? When do you need meiosis and when do you need mitosis?
Reproduction for meiosis and repair and growth for mitosis.
Yes, meiosis you need it only during reproduction, right? Certain time in life, which is the reproduction part, when you need to produce the sperms and eggs, right? Which is something occasional that happen, not every day, not on a daily basis. Mitosis, every time, all the time, all the time. You mentioned the reasons for the mitosis, whatever, any one of those reasons, but it's happening all the time. How many type of spring cells? Is it
Haploid for meiosis.
is it haploid or diploid? Here
Haploid,
and here.
diploid, yeah.
How many cells do you produce here?
4. Oh no, there's 2 divisions.
I mean, how many? Yeah, how many type? How many cells first?
4,
4, and here
2,
2. How many rounds?
2 rounds. 2 rounds.
2, and how many rounds here?
2.
And here 1. It's all these steps are just 1 round, isn't it? Starting from the prophase all the way to cytokinesis, this is just 1 round. The other one is 2, right? Line up, separate, cytokinesis. Line up again, separate again, cytokinesis again. It's 2 rounds, right? So here it is again. Remember, we call this N, and this will fertilization. We produce a zygote with 2 N, and this process to produce the gametes is meiosis, and then this is mitosis. And here's what we just talked about. Okay, you can do your own study guide or study aids by comparing the meiosis to mitosis. But did we get it? Did you get the mitosis versus meiosis? Is that very clear? You got it? Did you do the lab for this? Or not yet? No?
Yeah.
You didn't do it yet. Okay. But a good part of it, as far as I remember, will count on this. So understand this. Okay. Now the next part is variation. Variation. You know that everybody looks different, right? Even twins, identical twins, if you do mapping of the DNA, you will find not just the DNA, you will find some differences anyway. So variations, how the variations happen? Variations will happen during sexual reproduction in 3 different processes that we will cover in details. These processes are called independent assortment, random fertilization, and crossing over. Okay? And this will explain billions of people look different. Even no matter how much you think they are very similar, there are differences, right? How did you get all these difference? Millions and billions. How did you get it? 3 things: independent assortment, random fertilization, and crossover. So the first one is independent assortment. How does that lead to variation, and what does it mean? Right now we're talking about the sexual reproduction, okay? We're talking about gametes. I'm not talking about mitosis. I'm talking about the gametes, sexual reproduction, okay? How we produce different organisms, or in human case, how can you produce humans that looks different? So we're talking about independent assortment. Independent assortment mean assortment that's independent to each other. Assortment means arrangement. So you arrange different things that's independent to each other, and you will understand it. Remember that we have meiosis 1 and meiosis 2? Right? In the sexual reproduction, we have meiosis 1 and meiosis 2. You're repeating, right? During meiosis 2, do you remember this part, first part, when you line up here?
Mm-hmm.
So, which one will go with which one? You don't know. Like, this is called random assortment, okay? For maternal and paternal. Like, this one have 23, this one have 23. Which 23? Okay? Like the parents will have a lot of chromosomes, okay? Which ones will come to this specific sperm? You don't know. I know it's 23 anyway, but which 23? You don't know. Okay? Same thing for the mom. So this is called the independent assortment. And if you talk about it, it's 2 to the power of 23. That will give you 8 million possibilities just for this one. Okay? Why? Because you don't know how it's going to be arranged. This with this, or this with this, or you flip it, right? You have all 23 and they can be arranged somehow. Okay? So that will give us the independent assortment, and you don't know which one will be assigned to which sperm and how the chromosomes will be arranged. So independent assortment means the chromosomes will be assigned randomly. Yeah, you don't know. Remember, they're going to separate and then they are going to join again. Which with which, we don't know. This is random. Random fertilization by sperm and egg. Which sperm with which egg? You have no idea, right? Like, you know that sperms compete on the egg, and one of them, normally it's one. Can it be two?
Yes.
It can be. Two will lead to what?
Twins.
Twins, identical twins. Can two enter to the egg at the same time? It's rare, but it can happen, which is twins. Okay? But normally it's just one. Which one? There are millions of sperms. Which one? You don't know. Which egg? You don't know. So this is another round of random fertilization now. Randomness. So the first random was independent assortment. How are you going to arrange? You don't know. There are a lot of possibilities. This with this. This is carrying genes, this is carrying genes. Why this and this and this? Why not this and this and this? There are a lot of possibilities. And this is the second one, which is now will give you like 64 trillion possibilities because of the random fertilization. You have millions of sperms. Which one? You don't know. Right? 8 million per ejaculation. There are millions of sperms per one ejaculation from males. So you don't know the chances. Which one is the strongest of all? And it can be, a lot of them are strong, but the first one that arrives, we don't know. So that's why it gives us trillions, okay, of possibilities. And that's why no matter how many people we have on Earth, they will never look exactly the same. Okay? The third one is crossover. What crossover means? Here is what's happening. This is a pair of chromosomes. Okay? A pair of chromosomes. This is this one chromosome that's coming from the mom, the maternal one, and this is the paternal one. They are supposed to, are we following? The sperm have 23 and the egg have 23, right?
Yeah.
They combine. Which one will come with which one? You don't know, right? But when they come together, they are supposed to come together and just stay like a pair of chromosomes like this, right? That's it. But this is not what really happened. What really happened is, occasionally, sometimes, part of this chromosome will leave and jump into this one and exchange. We call that crossover. Look at this. It's supposed to be one red and one blue, right? While they are together, a part of this piece of this detach and jump and go here in exchange from the other side. Look how this looks like now. It's not pink red anymore. It's mainly pink, and there is a piece blue that exchange with this. So you're crossing over. Did you get this? What do you mean crossover? You're crossing over from one chromosome, piece of this chromosome, cross over to another, to the other chromosome in exchange for part of it. So if you put all these three together, that will give you that randomness that we see. Now, during meiosis, can something happen during meiosis that can be wrong, that's not supposed to happen? Sometimes it can happen. Occasionally, it's rare, but it can happen, specifically for older people. If you are pregnant at the age of 35 is not that old, but starting from 35. The older, the more chances, right? 40, 45, 50, the older, there are more chances that something wrong happened to the chromosomes, okay, during meiosis. Something happened during meiosis. So here is a scenario. It's telling you Jason and Laura are pregnant with their third child, but they are over 35. So they wanted to do a test. They say, okay, we are over 35, so we wanted to make sure that there is nothing wrong. We know that when the older we get, the more chances that something happened. We need to be tested and see what's wrong, if there is something wrong, okay? So they do something called amniocentesis. Amniocentesis is two parts, or amniocentesis. Amnio is amniotic fluid, that fluid that's around the fetus, okay? Centesis means, or synthesis means withdraw, like you put a needle and you withdraw. So amniocentesis means you draw some or part of the amniotic fluid, okay? And when you draw this part of the amniotic fluid, you can take a picture from the microscope and show it to them, which we call that karyotyping. Do you remember the karyotyping? Photographic description of the chromosomes. And when that happened, they did that and they found 47 chromosomes. Is that normal?
No.
It's not normal. It's supposed to be what?
46.
46. And there is an extra one, right? In that fetus, there is an extra one. So there is something wrong. What happened, most likely, is an abnormality that's called nondisjunction. Nondisjunction. And nondisjunction is, what's junction means? Two things together, right? Disjunction means separate them apart, right? If that doesn't happen and they stay, non-disjunction. Did you get this? Here is what I mean. Remember when you arrange the chromosomes along the equator like this, okay? And then the threads come and separate them apart. One set go to this side and the other set go to the other side, right? What if one of the pairs didn't separate? So two of them will come in one side and the other side doesn't take anything. Did you understand my idea? Did you understand what happened here? So if that's the case, one of them will have extra and the other one will have less. So instead of, if this is a fetus that's coming from 23 from the mom, 23 from the dad, it's actually 24 from the mom or the dad and 23. So the total is 47 I mean, yes.
What would happen to the baby?
They will have abnormalities, and depending on what type of abnormalities, I will talk about this, but it's called a trisomy. So they will have 23 pairs, but one of the pairs is not 2, it's 3. So the total is 46, but this will lead to abnormalities, this will lead to issues, the mental status will be affected, the muscle, the heart, a lot of things will be affected, and I will talk about this. But just make sure that you understand this, okay? I wanted to make a sperm and that contains 23. Okay? You're supposed to do that. So here are the chromosomes. Separate, and then you do, the next time you go like this. Separate. Okay? What happened here is, or even this is 23 pairs, okay? Here are the spindles or the threads. What is this? What is this called? These are the spindles. What is this? Centromere? Centromere. So the spindles are going to pull. And this set's supposed to be here and this set's supposed to be here. Is that okay so far? So here's what's going to happen. And those, non-disjunction, they didn't separate, and it will be like this. And here, nothing. See this? So this, if you put them together all, that will be 24, and this will be 22. Did you get the idea? Did you understand this?
Yeah.
So instead of 23, now it is 24 and 22. If this 24 go with 23 from the mom, how much? 47. Did you get the idea?
Yeah.
Which pair didn't separate? This will give you different conditions, okay? But the most common one is 21. 21. Why? Statistically, the one that can have a chance to the pair that have a chance that the two chromosomes stay together more to be 21, number 21. So from 1 to 20, they separate, okay, everything is separating. When you come to 21, this is most likely the one that does not separate, okay? So here, if you translate that into numbers, it's supposed to be 1n. What's the 1n?
23.
23. And you call it haploid or diploid? 1n is 23 haploid, right? And another 1n haploid. They come together to make the 2n, right? And instead of just being 2n, which is 46, it will be 2n plus 1 or minus 1, which is this. You don't know which sperm is going to fertilize, okay? So this is called non-disjunction, and the chromosome will not be normal. So here it is, if you translate that, normally you have 2n, here it's not 2n, it's 2n plus or minus 1. And here is the most common that I talked about for non-disjunction, which is called trisomy 21. What's tri means? 3. What 21? 21, what are we referring to? The pair number? The pair number what? The pair number 21. Is that autosomal or the sex chromosome? It's autosomal or sex chromosome. Number 21. Autosomal. Which one is the sex chromosome? Number 23. So number 21 is autosomal, right? Okay. So this, you see that this is what I described here? Let's say this is number 21. So we have a lot before that, and everything is okay except for this one. Okay? So this is the most commonly one that does not separate, and we call it trisomy 21. Why? Because the first pair is 2 all the way until 20. 21 is not 2. 21 is 3. So we call it trisomy 21, and trisomy 21 will lead to, this is what you were asking about, trisomy 21 will lead to something called Down syndrome. And you need to understand that. Down syndrome. So what's a Down syndrome? Trisomy 21. What's the reason for that? What caused it? A condition called non-disjunction. Are we following? Trisomy 21 non-disjunction. Look at this. What do you call this? What I'm looking here. What's this dark thing here? What is this? Huh? Karyotype. Yes. What you're looking at here, this is called karyotype. This picture that you're seeing. You know how they do it? They take picture from the microscope. While looking, there is a button like this. They press at it, and it will give you a picture. Okay? So this is what you're seeing here. Look at number 21, 3 instead of 2. This is called trisomy 21. Look at this baby. He doesn't look really normal, right? He will be a little bit shorter, will have some mental retardation. This will be depressed, okay? This area here will be depressed. The eyes are going to be a little bit far away from each other, a little bit. So he doesn't look normal, and the brain is not normal. He will have some sort of mental retardation. Okay, this is not something that you need to know. Just know that Down syndrome is trisomy 21 because of non-disjunction. That's it. I'm just telling you in case if you're interested to know. He doesn't look normal. He's a little bit shorter. This is depressed. The eyes are a little bit far away from each other, a little bit. The mouth is kind of smaller a little bit, and most importantly, mental retardation.
What's the average lifespan? Or is it?
I'm not sure about that. It depends on the condition, because it's not one thing only. The heart will be affected, the brain will be affected depending on the conditions. But as far as I know, it might be like 20, 30, something like that. I'm not sure about that. It might be more than that. Okay? What if the non-disjunction happen in the sex chromosomes? Which pair is this?
That's why.
Which pair? 23. 23 is supposed to be 2, right? 2 chromosomes. Either XX and XY. So what's happening here is same scenario here, but instead of from 1 to 22, it happened to 23. Are we following? This is pair, I'm talking about pair number 23 only now, okay? So here you have Y and X. And here you have X and X, okay? This one didn't separate. So this sperm contained XY and this sperm contained what? Nothing. Right? It doesn't contain any one of those. Both of them, they are supposed to separate, right? They are supposed to separate. So they didn't separate. And this one is, it's okay, X and X. Here is 1 and X and here is 1. Can you see this? If this sperm fertilized this ova, what's the result? X, X, Y. Isn't it? This and this will be the same thing, isn't it?
Yeah.
Okay, how about this? Where is the other X? 1 X from here. What from here? Nothing. Nothing. She was just 1 X. Okay? So if the 6 chromosomes did not separate, you will have one of these conditions. Okay? X, X, X. Depending on which one did not separate. Okay? X, X, X. We call it the triple X syndrome. X, Y, Y. We call it the Jacob syndrome. Jacobs. X, X, Y. This is called Klinefelter syndrome. You need to know this. XO is Turner. This is what I was talking about, which is like XO. Are we following or not? Are we following? So if that non-disjunction happened to the 6 chromosomes, which is pair number 23, you will have different conditions, and you need to know this. Okay? So triple X syndrome, this is obviously a female. Okay? And it's not going to be a horrible thing. It's just a female that's taller and learning disabilities a little bit, but she will survive. Okay, XYY, this is male. Same thing, a little bit taller, a little bit learning disabilities, nothing much. XXY, this is an issue. Okay, XXY is a male that's sterile. He will never have kids, children. Testis will be small, and he will have some feminine characteristics, like he will have a breast, okay, and the configuration of female kind of because of the extra X. XO is a Turner syndrome, and this is a female that's also sterile, and she have immature 6 organs. Basically, she looks like a boy, kind of. Okay, she have the reproductive system, but underdeveloped, and she looks like a boy, like no breast, and the other organs will not be fully developed. Okay. In order to know which is which, just to make it simple, if I ask you, like, if this is any one of these 4 conditions, is it a male or female? The only thing that you look at, Y. Y, male. No Y, female. That's it. Okay. So does this have Y?
No.
This is a female. Triple X. Does this have Y?
Yes.
Male. Does this have Y?
Yes.
Male. How about this?
No.
No. So do you have Y or not? That's it. Okay. The last one here is called Turner syndrome. Okay, Turner syndrome, and this is a sterile female, just like the Klinefelter syndrome. The Klinefelter syndrome is a male that looks like female because of the extra X. And what was the reason for all of this? Non-disjunction of which pair? 23, which is the 6 chromosomes. Okay? Is that clear?
Wow.
Okay, so inheritance. Next part is inheritance. Okay? When you look at children, they look to a good extent like their parents. Families looks like close to each other, and you can tell. You can inherit certain things, and you don't inherit everything. And this will take us to the inheritance with Mendel. Okay? So what's definitions first? What's hereditary versus genetics? Hereditary is the transmission of a trait from one generation to the next. Transmission of what? Of a trait. What is the trait? The trait is anything that you can see in a person. The hair color is a trait. The eye color is a trait. Are you tall or short is a trait. Anything that you can describe is called a trait. Clear? The trait of green eye, the trait of blue eye, the trait of yellow hair, the trait of black hair. Is that clear? The trait, right? So this is hereditary, is transmission of traits from generation to the next generation. Genetics, on the other hand, is the study of that hereditary. If you're studying that, we call that genetics. And this will lead us to Mendel, which put the principles for inheritance. And we will discuss that. So here is characteristics. What's the characteristic versus trait? Characteristic is you're describing one thing. Trait are the different possibilities. Example, the character of eye color. I didn't determine, right? I didn't say what color. Just generally speaking, the eye color is a character. The hair color is a character. And when you describe the details, now you call it traits. Okay? So if I'm talking about hair color, what do you call this? Character. Yellow hair. Trait. Black hair. Trait. Did you get the difference? So when you describe, generally speaking, we are talking about the eye color. This is character. In that character, when you describe it, green, blue, black, brown, this is called trait. Each one of those is a trait.
Oh.
Now alleles, this is the individual unit of inheritance. The unit of inheritance, okay? And they come from genes. So alternate forms of a particular gene is called alleles. What do you mean by that? Here is what I mean. The gene of the eye color. The gene of the eye color. Okay? That gene of the eye color. Where did you get it from? From your dad or from your mom? Both of them. Right? So you will have one allele from here and the other allele from here. Okay? Which one will give you the color? It depends. We will talk about this. But generally speaking, you're having alternate forms of a particular gene that's called alleles. Okay? Remember that alleles is not trait. Did you understand this? Trait is what you see with your eyes. This is a green eye, this is a blue eye, this is a trait, right? Allele is the gene for that trait. Clear? Is that clear? For alleles. Okay. So if you look at this, this is giving you the two different alleles. Let's say blue is from the father and the pink or that red is coming from the mother. Okay? Can you all see this? Mm-hmm. Those two? Obviously, this pair is coming one from here and one from there, right? You take 23 from here and 23 from here, right? So each pair is half and half. You take half from here and half from here. Okay? So if you look at this, which one is the father? It's the blue. Okay? And this is from the mother. If you look at the father, this is the allele for the eye color. And this is B, which is uppercase. Okay? And this is the allele also for eye color, and it is also B, but lowercase. What's the uppercase and lowercase? Uppercase is usually the dominant, which is when there is a difference, it will take the upper hand. Okay? We usually, and when we talk about this, when we talk about inheritance, when we talk about these alleles or genes, when you put uppercase, it's dominant, lowercase, it is recessive. Okay? And obviously, if you have two uppercase, okay, no problem, right? like uppercase black hair, uppercase black hair. Okay, both of them are dominant. We don't have an issue. But the thing will be, what if I have one uppercase and one lowercase? Which one will take the upper hand? The uppercase. That's called the dominant. Okay? If the two alleles are different, we call that heterozygous. Heterozygous. Hetero means different. If it is the same, we call it homozygous. Okay, so this is homozygous. And this is heterozygous. We are going deeper. Okay, so you need to follow with me. And if you have a question, let me know anytime. Okay? So are we following so far? We're talking about a baby. And I'm talking about this pair of chromosomes specifically that have the eye color. Okay? This pair have two alleles. I'm talking about the eye color. Two alleles of the eye color. One from the father and the other one from the mother. If both of them like each other, I call that homozygous. Okay? If one is different than the other, I call that heterozygous. Is that clear so far? If it is uppercase, let's say B is for the eye color. Okay? B is for the eye color. So if B uppercase and B uppercase, homozygous. B lowercase and B lowercase, homozygous. If they are different, one uppercase and lowercase, irrespective which is which, we call that heterozygous. Is that clear so far? Heterozygous. Now, genotype versus phenotype. Phenotype is observable trait, what you can see with your eyes. Okay, and genotype is the genes behind the phenotype. Okay, so you are light colored, for example, or dark colored. Is that something that you see with your eyes?
Yes.
This is phenotype. Which gene caused you to be light colored or darker colored? This is a genotype. Did we get it? The gene that's behind the phenotype is called genotype. Is that homozygous or heterozygous?
Homozygous.
Homozygous. And this?
Homozygous.
And this?
Homozygous.
They are all homozygous, right? And what's the F? This is dominant, and it is an allele, a dominant allele, and another dominant allele. These are two alleles here, and both of them are lowercase. So this will take us to dominant versus recessive. If you only if you have heterozygous. If both are similar, both of them uppercase or both of them are lowercase, what do you call it? Homozygous. If one is different than the other, heterozygous. Okay? So if it is homozygous, we don't have an issue, right? Yeah. Both of them have the same thing, so you don't have an issue. There are no possibilities. Both of them are the same. But the possibilities will happen if you're heterozygous. Okay? And if it is heterozygous, the dominant one will take the upper hand, and it is the one that's going to determine what's going to happen. Okay? Like if you look at this, for example, this is Mendelian inheritance. It will become more complicated later on. Okay, so follow with me. This is a simple form, which is the Mendelian. This is the simplest. So here we have the purple color. This is P uppercase. So this is dominant or recessive? Dominant. And this is the lowercase, which is white. Recessive. So if we have those mixed, which one will dominate? The purple. Okay? This is if we're talking about it from the Mendelian point of view, which is the simple way. Now, prediction of inheritance pattern. Can you predict what's going to happen or not? Most likely you can, and there is a way to predict, and we will talk about this. But here is something like PTC. PTC is a chemical that's very bitter. If you taste it, very bitter. But some people, when you give it to them, they taste it, it doesn't taste anything. Like I gave it to you, what do you think? It's very bitter. What about you? It's very bitter. What about you? Nothing. Okay? So this is something that is dominant. So if you have the dominant from the dad and the dominant from the mom, it will taste better, right? What if you have one uppercase and one lowercase? You'll still taste it, right? What if you have two lowercase? No. When I give it to you, say it doesn't taste like anything. I'm not tasting anything. Okay. So this is just a description or an example of that pattern of inheritance. Punnett Square. Are you familiar with this?
Yes.
Punnett Square. Okay. You should be, because you will see questions on this. Okay. You can see the Punnett Square itself, or you can at least imagine how it's going to be. Okay. So in this Punnett Square and inheritance in general, you will have, if you have generations, the parents. This is called the P for parent, and then each generation will be referred as F1, F2, F3. So this is the kids, the grandkids, and so on. Okay. Obviously, Punnett come from the name of the scientist, just like everything else. Most of the stuff come from this. So here is the Punnett Square, and if we're talking about this is the dad or the mom?
The dad.
This sperm, right? So this is the dad, and this
Mom.
the mom. So if we're talking about something like this, okay, inheritance of the color of the dogs, a dog like this, okay, the black color is dominant, which is the uppercase B. Okay, the chocolate color is recessive, and we call it B lowercase. Okay, are you familiar with this? Punnett Square. So what would be this one?
That would be lowercase b.
Uppercase and lowercase.
One uppercase, one lowercase. Which one is dominant? This, which is the black, right? So he will be black, right? How about this? How about this? All of them will be like this, right? This is a very simple, straightforward, okay? But the point here is we call that monohybrid. Monohybrid mean I talked about one thing only, color of the color of the dogs. Did I mention anything else?
No.
Just the color, right? Black, chocolate, color. Later on, we will be talking about more than one thing at the same time. Okay, but we just remember that we call that monohybrid. Monohybrid, like you're combining two things. Hybrid means you're making a hybrid, a sperm with the ova. This is hybrid, right? Monohybrid means I'm talking about one characteristic only, one inheritance only. Okay, can we talk about more than that? Like this sperm have the black color and have the mental retardation and have the color blindness or whatever. We can talk about more than one, but I'm talking about one now. As long as I'm describing one thing only, we call that monohybrid. Okay, so this is the chocolate one. This is dominant or recessive? Recessive, both of them, right? And this? This is dominant and recessive. But what's the color? Black. Why? Because the dominant take the upper hand. The dominant is the one that determines. Okay? Can you tell me the chances here?
It's
This?
50. Well, that's 100%. Let's see if I'll do that.
But just tell me the combination.
25.
Uppercase,
Lowercase.
lowercase, and here
Uppercase, lowercase.
same. Here
Lowercase, lowercase.
and this.
Lowercase.
So it's 50-50.
Yeah.
Black versus chocolate, right? This is in this scenario. So how does that happen? Like how did you build that Punnett Square? Because the chromosomes, remember this is a pair of chromosomes, right? And the allele, let's say this is the allele for the color of the dog, and this is the other allele. Okay, both of them together determine what the color of that dog, right? So now during meiosis, you're going to separate those. So this one will come with B uppercase. Let's say if this is a heterozygous, and this one will get the B lowercase, right? They are going to separate from each other, and we don't know which one will separate with, will combine with this, and which one will combine with this. And this is just the basis of the Punnett square. Did you get how it happened? Which is the segregation, the law of segregation. So the law of segregation is state that the two alleles for a character, they separate or segregate during gamete formation. Okay, here is one allele and here is one allele. They used to be together, but they separate, and each one go to one of the gametes, and then they are going to recombine when we make the zygote, like this, black, but it's B and P, and they are going to separate. So this will give us the chances. Here is one, and here is one, and the same thing for the mother. And we are going to recombine them in 4 different possibilities. Can you use that test cross to determine the genotype? Can you know what kind of genotype? Like this. This is black, okay? What are the possibilities here? What do you think this is?
It can be either or, the capital or the lower.
Right? Is it uppercase?
It can be either or.
It can be all uppercase or lowercase, right? Because it's the black is dominant, so we don't know, right? But this is chocolate. For sure, it is what?
Lowercase.
Both of them are lowercase, for sure, right? Otherwise, it wouldn't be. If it has 1 and 1, it will be black. If both of them are uppercase, it will be also black. So if you see it recessive that's showing up, the only chance is both of them are recessive, right? And this, we don't know. The only way to know what is this is when you match it and show me the offsprings, and I will tell you what was this. Okay? So here are the chances. If you assume it is black BB, so this is in the previous one. We're trying to solve it now. Are we following? This is chocolate and this is black. The chocolate is B lowercase, B lowercase. Is that okay?
Yeah.
The other one, I don't know. It can be both of them uppercase or it can be mixed. I don't know. How to know? If all of them came black, then it was BB. Uppercase, uppercase, right? Make sense? If it come like this, it was mixed. It was uppercase, lowercase. Can you tell the chromosomes of the parent when you see the offsprings or not? This way, right? So this is only you're showing me a black dog. Tell me what kind of alleles and what are the chromosomes. I don't know. Show me the offsprings and I can tell you, right? It's black. I don't know. Black B uppercase, uppercase, or uppercase, lowercase. Show me the next generation and I will tell you. The next generation are all black. It was uppercase, uppercase. Half and half. It was uppercase and lowercase. So can you tell? Right? Okay. Okay, and we will stop here and we will continue next time.